Okay, hi. So, yeah, thank you for inviting me. So I'm a bio designer and an architect from Toronto, and I work at ETH Zurich. And I work on developing living and dying structures at architectural scale with the goal to augment objects of design, products, buildings with some sort of biological functionality. So I'm going to start with this story that I studied in my first year of architecture school. So this is an image of Gilgamesh, the king of Uruk, one of the first known cities, which is founded about 4000 BC in Mesopotamia. And Gilgamesh and his conquered companion, Enkidu, are slaying Humbaba, who's a monster that protected the sacred cedar forest. After they kill Humbaba, Gilgamesh cuts down the sacred trees to build gates for his city walls. After a few more transgressions, Enkidu is killed as punishment by the gods, and when Enkidu's body starts to rot, Gilgamesh, who's fearful of death, casts the corpse outside of the city walls and makes a statue of Enkidu inside the walls to immortalize his friend. So Gilgamesh is taught to us as young architects as the first great builder, who, terrified of the uncontrollable wilderness, uses his cunning to build a city and tame his surroundings and separate culture from nature in the belief that he can build his way to immortality if the statues are tall enough and the walls are sturdy enough. And he's the first great hero of Western literature. So 6,000 years later, we see the legacy of Gilgamesh's attitude in how we build today, when we kill nature's protectors to extract the resources we want and build the cities we want. We know what happens when we approach the world like this, when we try to conquer the wilderness and all of nature's monsters. We often end up accidentally deleting the processes of protection, resilience, and regeneration that are paired with these monsters. So the thrust behind much of my research in the last 10 years has been trying to revive some of these monsters and learn how to work with them as integral parts of the built environment, recognizing that the built world is part of the larger natural one, and we have to design it as such. This means to find new and also very old ways of making and breaking things that are regenerative rather than extractive, and learning how to use materials that we can build with that will service the natural environment. And crucially, it means to try to understand the shift in design thinking required to work with these materials and processes that have a certain degree of agency and currently lack the level of industrial predictability that we're used to as architects and engineers. And so the research is focused on understanding and applying the design principles of living systems to create sustainable, intelligent materials for the future. This is partly with the ambition of reducing environmental impact of construction, but is also in a large part to explore the new design methodologies that come from working with natural systems. So what form do these monsters take, and how can we live with them in our built world? And how do you incorporate them? How do we design with them? Because in the end, we need these monsters in order to access certain desired biofunctions. So things like regeneration, which often comes with organic decay. This is an image of a set of Guan rock paintings in Western Australia. They're about 30,000 years old. And there's a starting point for some of my previous work on decay. And so these are very vivid paintings despite their age and exposure to the elements. And that's because the original pigment, which was scratched into these cave walls, have actually been decomposed and eaten by a symbiotic community of black fungus and red algae, so a lichen, that's colonized the lines of the original drawing. And the paint is rendered then for thousands of years in this self-renewing biological pigment. So it's a model of what our built environment could be if it leveraged biology's power to repair and replicate, creating this longevity out of responsivity and renewal rather than the static robustness that many industrial systems use. So as a precursor, these are pieces from Agahoya 1 in 2017, which I worked on when I was involved as a research assistant at the MIT Media Lab in the group led by Neri Oxman. It's a biomaterial project, and so we had made this 5-meter tall pavilion and a library of artifacts out of 3D printed chitosan, cellulose, and pectin, which are the most abundant biopolymers on the planet, and they're water soluble and biodegradable. So the material system is environmentally responsive and can grade it through our 3D printing process, which is by varying the proportions of the 3 main ingredients, so putting different amounts of different things in different areas. Then you can change the quality of the print, right? So you can get a vast range of mechanical, optical quality from this and control things like color, opacity, strength, and flexibility. Because this is a system that's biocompatible and collapses eventually with the application of water, we also had the idea that we could tune the speed of degradation in water through material composition and toolpath strategy. So like denser paths, more material, more robust material composition will degrade more slowly than looser paths, less material, weaker compositions. But sometimes these denser paths actually also make the material more responsive. You get more deformation. And so you could also tune, you could harness some of this responsivity and use the chemical tunability to design movement using something like humidity as a stimulus. So with kind of this material system that I had been working with at MIT, and the ideas of like harnessing the decay process, I did a residency at Ginkgo Bioworks in Boston. So Ginkgo is a synthetic biology company that engineers organisms or their byproducts for use in cosmetics, drugs, industrial applications. And they also support a designer in residence every year. And so the theme of my year was how to design a world without waste. And my response was that if I know everything I design as an architect will eventually become waste, can I design that waste intentionally, not just as something that will eventually break down, but also as an input for future renewal? And so design by decay, decay by design, it's a series of artifacts that exhibit design decay. And so we use biological entities like enzymes, bacteria, and fungus as agents of material transformation throughout an object's life cycle, not to destroy it, but to reorganize it into different forms that might still be useful or desirable in some way. Could we spatially and temporally guide where and when decay might occur? Perhaps through depositing the enzymes or microbes, certain nutrients or certain inhibitors in different areas at specific times, at specific concentrations. So this study used enzymes, which were removed from the organism, to cause things to fall apart. And so enzymes are the powerhouses of our metabolism. In decay processes, they often break down complex molecules down into digestible parts that can be used as feed or for building blocks for other organisms. And I would say this is the most mechanistic study I did at Ginkgo, where because the enzymes are divorced from the organism that produced it, we could kind of control this a lot more specifically, I guess. So we have different types of enzymes that are applied in very specific concentrations for very specific amounts of times to certain areas in my biocomposites, and they'll actively eat away holes or cut away lines in the material. This was a study I did with green mold, which is a trichoderma variety, and a black mold, Aspergillus niger, to co-colonize some maple carvings that are computationally designed with an expanded surface area so that you have more substrate and interface for the microbes. And we did 4 identical carvings made with a CNC, and then you see some of the tests became quite contaminated and other ones became kind of like green and black paintings. And some of them had to be thrown out because they became so gross. And so what's interesting, though, is I think the white one actually got contaminated with a mycelium. And so the wood block becomes soft and spongy, but the mycelium that replaces it gets stronger and stiffer with time. As the decay work led to this installation that was done in Toronto for the Rhubarb Theatre Festival, trying to take the work that I was doing inside of the wet lab and upscale the processes I was working at. So we inoculated a bunch of fallen trees. So we gathered a bunch of fallen trees from different forests around Toronto, and then inoculated them with different funguses. So trichoderma, white rot, other mycelium, and then we made a bootleg version of a laminar hood inside a theater, and then set up this installation. So the theme was how to regenerate practice after 2 years of the COVID pandemic, and what do you leave to decay and what is left to rehabilitate afterwards. And so the work started its process of decay in the theater, where we had built these weird pressurized tents for the pieces. And then they were installed for this theater festival where 10 different performance artists were asked to use the work as a setting for their own performances. And they were asked to respond to the processes of decay that were kind of surrounding them in the theater, with the context that this was their first return to public theater after the COVID lockdowns. And then after the festival, we had to find a suitable resting spot for the structures so that they could eventually grow old and die and be recycled as fodder for the new organisms. So we returned the stumps to the same forests that they originally gathered from. And so we're kind of watching nature reclaim them for her own purposes over the next few years. So a shift, okay. So my main research now involves carbon sequestering bacteria, and I'm trying to learn how to integrate them into building facades to make use of the built surface area of a city to capture carbon. So this is Xenococcus PCC 7002. It's a marine cyanobacteria that lives in salt water, and it's capable of something called microbial-induced carbonate precipitation. And so this is its metabolism. So PCC 7002 does this through photosynthesis. It consumes CO2, produces oxygen in return, and as a byproduct, it also produces carbonate minerals, which creates this dual carbon sequestration mechanism. So one through biomass, through photosynthesis and growing, and the other way is through these minerals that are a potentially long-term form of carbon storage. So this phenomenon is seen in nature a lot, in stromatolites, and they're formed in these shallow salty water pools where cyanobacteria gets caught in microbial mats and sand. And then the trapped cyanobacteria consumes CO2, produces oxygen, and then it causes the precipitation of the calcium carbonate crystals that bind the sand grains together. And then they harden these structures over thousands of years. So the structures are ancient, but they're still processing CO2 today. And in terms of impact, billions of years ago, the planet used to be covered in stromatolites, and the cyanobacteria in them are what made the planet go from a high CO2 environment to a high oxygen environment and lowered the planet's temperature as a consequence. So the first few years of my PhD, I spent them trying to make these small artificial stromatolites with my collaborators in the macromolecular engineering lab. And so this is a lab mostly of material scientists, and they focus on working with hydrogels for cell encapsulation. So we did all of the fabrication through additive manufacturing processes because we're trying to get quantifiable high surface area structures that have the potential for more nutrient, light, and gas diffusion. So the tests range from prints that are made out of pure polymer, where we encapsulate the cyanobacteria in like this hydrogel and it's bathed in a nutrient media, to prints where I start to use sand as a filler as a filler material between the hydrogel and the cells. And so this is what came out of one of the small studies, which is this paper that recently came out in Nature Communications, and it's on the carbon capturing capacity of this system. So this small little pineapple lattice, which is less than 2 centimeters tall, and we call it Josh. We kept it alive for 400 days in the lab, and the amount of precipitate it made was quantified and analyzed. And so what's really cool is that the precipitate is deposited primarily in the hydrogel scaffold, so you can make these little carbonate skeletons that are in the shape of the print, which is kind of, this is not Josh, but yes, this is where you burn off the polymer and you just have the carbonate skeleton left at the end. And what happens is the carbonate skeleton also mechanically reinforces the print, so it makes it stronger and harder. And so you can see with Josh, he's pretty floppy at the beginning, and then as he gets older and more reinforced, he starts to stand up. And then the amount of precipitate that was created was measured via this method called thermal gravimetric analysis over the 400 days. And so my collaborators, Ifan and Dalia, they're able to show that this structure is sequestering about 26 to 33 milligrams of CO2 per gram of printed material. So this seems very small, but it's a comparable value to existing industrial methods of carbon capture, for example, using the chemical mineralization of recycled concrete, which is commonly used right now. And it's actually much higher than most biological means of carbon capture. And it happens with mild conditions under fairly passive, like low-intensive biological means. So the question for me as an architect then is how to do this at scale. Like all the tests we had been doing for 2 years were very interesting, especially the ones using volumetric printing or DLP, but they're so small, like they're all less than 2 centimeters, and they're extremely expensive. So I spent a few years making a larger platform, and my colleagues and I have developed this biofabrication platform where we can 3D print living structures at a larger scale out of granular material with microorganisms embedded into them. So the system is organism agnostic in that we can try different species to gain different biological functionalities within the prints. So these are the growth curves of 3 different bacteria that we tried, and they're mapped against the certain products of their metabolic activity that we are interested in. So for PCC 7002, the photosynthetic biocementation process is always accompanied by a rise in pH, and so that's what we're measuring. For S. pasteurii, which is the common bacteria used to consolidate soil, people measure the amount of a certain enzyme activity that results in the biocementation, and you map it against its growth. And then for photokishitani, which is a glow-in-the-dark bacteria, we're measuring the amount of photoluminescence that's given off as a function of the amount of calcium that's in the media. And so by understanding what these organisms need to survive and the time scale of when this activity happens and how long we can maintain them, we can integrate these different species into large-scale 3D printed objects and make them capable of things like biocementation, bioluminescence, carbon capture. So these are prints that I did with my students and some collaborators, and then we try to, they're done with different inoculation populations, different print media. So this is kind of the process for PCC. So different type of population at the beginning, different print substrates, incubation conditions, and timeframes. So much of my work is focused on the development of scalable processes that we can test outside of the wet lab. Some of it is successful and a lot of it is not. So this kind of shows the jump in fabrication scales that we're able to achieve every year with the platform. Compared to my tiny, tiny little prints in 2022, the first medium scale print I did with my student Nijat is about 30 centimeters tall in 2023. And then with my 2 thesis students last year, we were able to get to this 1 meter tall arch using different topology optimization strategies. Actually, this was done with Mark Tan as a thesis coordinator. So the reason we have to use topology optimization on this is the material is extremely weak and also fairly heavy. It's very low compressive strength, like 2 to 3 MPa. It's a density similar to lightweight concrete, and it has no tensile capacity. So we have very strong material limits in the fabrication platform, and that dictates things like how long an unsupported strut can be, what the diameter to length ratio should be, and also how large or dense the print can be before it collapses under its own weight. So like this print we did, which is our largest test sample, about 1 meter long and 40 by 40 in cross-section, it took about 5 people to lift it. And then we have to take this very weak starting state into account when designing this material with the intention that the material should strengthen with time as it biocements, but that time frame may be years or decades. So we were given the opportunity to showcase the platform for the Canada Pavilion at this year's Venice Biennale, and really tried to push the limits of what we could fabricate with the system. So the project is called Picoplanktonics, and it's done in collaboration with 2 universities, so Toronto Metropolitan University and University of Toronto as the exhibition partners, and then ETH is the research partner. And we also had Clayton Lee, who is a Canadian performance artist based in the UK. And the project is an experiment of what would happen if we could take this material outside of the wet lab and at the scale of interior architecture. So when we thought about Venice as a site, 2 things come up very quickly. These very common images of Venice underwater, and then Canada Pavilions placed in the Giardini, very close to the lagoon, as well as where these photosynthetic biocementation processes typically occur. So our proposal for the Canada Pavilion was then to flood it with this shallow layer of water, about 10 centimeters, which is equivalent to the most frequent level of flooding in Venice, and then make a home for the living structures that we could also occupy. So we wanted to pay homage to the existing trees that are enclosed into the pavilion by referencing them in the design of these two kind of monsters, these little trunk pillars made out of a series of photosynthetic bioprints that are resting on a sand skeleton. So the Canada Pavilion it's a beautiful building, but it's notoriously difficult to exhibit in. It's not a white box, there's a lot of personality to it, and it's quite small. So it was important for us that the project not just look like a weird science experiment, which it is, but it's also something that we're trying to respect the original building myth. So if we're trying to incorporate nature and all her monsters into the built environment, what this means for the design process is that it's not just a human-centered act. And so when we're designing this environment, human occupation has to be weighed against our ecological goals and vice versa. So the monsters had to look appropriate to the space, but we had to actually change a lot of the building in order to accommodate them. So these prints are also the largest that we've ever made. So when we had received the commission, our largest print was about 30 centimeters tall, and we were proposing to make something over 3 meters tall. Along with the scale, the project's an experiment in the durability of the material and the viability. So taking it outside of the wet lab and letting the external world and all the microorganisms that are kind of part of all the visitors coming, as well as in the environment in Venice, interact with them for the first time. So this is a little trailer of how we made it. There's a lot of things I can't show because it's under IP. But then, so you can see cell cultures, material processing, some of the printing, toolpath planning, print extraction and processing, and then caretaking. So all the fabrication process happened across the ATH campus, in the Alive Lab, at ITA, and then in DBAUG. Then we had to pack all these prints and ship them to Venice in like 50 crates. With the skeletons, and the skeletons we installed first, and then we there's about 100 bioprints that we did, and so installation took about 3 weeks for the team, nonstop. Yeah, and so this is Picoplanktonics. It's a series of 3D printed living structures capable of CO2 capture that will grow and die throughout the 7-month exhibit. So the entrance of the pavilion has a series of these 9 incubation tanks where the conditions are much more controlled. We can regulate temperature, the amount of light that they get, and the humidity is much more constant. And we use them as a comparison point to these 2 main structures that are in the main exhibition space of the pavilion, about 1.8 and then 3.3 meters tall, and they rise out of this saltwater pool. Then at the end of the pavilion, we have this caretaking station. The space is basically a semi-controlled maintenance room. And so we have a space for all the salts and supplies that we use to replenish and monitor the structures. So we control things like pH, light levels, temperature, salt content, and gross contamination. We can't really control the humidity very well in this pavilion, which is actually quite a problem for the structures. And then the guests will exit, this is supposed to play, The guests exit into the courtyard and they encounter this outdoor garden. And you can look inside and see the structures, and there's a small little outdoor garden of these failed bioprints, like things that have fallen out or breaking apart, and we just leave these outside to the natural environment, and they can be poked at and stuff like that. So much of the project is about the amount of care required to keep this exhibit going. The structures have to be watered and monitored, and some parts will grow, others will fall apart, and other parts have to be replaced. And we really wanted to highlight the need for human effort in this endeavor, because it's not just about having a robot make it and monitor it. It's really about us making the time to work on this. And so we're really lucky in the Canada Pavilion because we have this team of docents who are young architects from Canada that get fellowships to come and work as the gallery assistants for a month, as well as do a project on Venice. And then we have a team of Italian tenants to help with this as well. We modified the pavilion to house the structures and kind of provide the warmth and light and humidity that the bacteria typically needs to grow. Also, I think the pavilion is actually quite appropriate for it because of the amount of natural daylight that we get inside the building. And so we have installed this large saltwater pond and a filter system inside the heritage building. We also installed grow lights throughout the space and environmental sensors. But it's really important to note that this is an open pavilion and not a wet lab. So what we're trying to learn from this is, what might work for cyanobacterial growth and what doesn't. Also, the project required the work of many different collaborators from various disciplines, from architecture, art, digital computation, fabrication, material science, and biology. I had 3 main interns plus an army of undergraduate students, as well as my collaborators in Hong Kong, Kuwait, Canada, all working on this. Yeah, and sorry, oops. That was, yeah. Yeah, so yeah, this is kind of the list of credits of everyone who's involved, from the Canada Council who funded the brunt of the project to all the sponsors, institutions, and team members. So yeah, I started with Gilgamesh in this lecture, and as this kind of symbol of the desire for mastery over the natural world. But I think most of us in the room will agree that this is probably not the best solution, and then perhaps believing in this kind of symbiotic relationship between the built environment and nature is possible if we kind of learn how to work together in this collaborative rather than confrontational approach. Okay, thank you.
Thanks, Andrea, for fascinating talk. And would anyone like to start with a question? I guess I'll have to go then. Yeah, I think it's for me, a very interesting way to end this symposium, because I don't think you mentioned the word artificial at all. But at the same time, I think when I see these pictures of these bacteria doing all these things that's not visible, I don't think it's too dissimilar from actually what artificial intelligence is trying to do. So I guess my question is, I think throughout the symposium we saw different modes or methods of artificial intelligence, and I think Adil this morning said, it's a beast that's growing twice as big, twice as fast every single day, and we're only merely trying to control a thing to see what comes out of it. But in nature, maybe all the permutations are already available to us. Maybe we're just trying to understand how they got there. How do you see your work evolving in parallel with this rapidly evolving artificial intelligence? And is there a way in which the natural intelligence could coexist, and how could they inform each other?
Yeah, I don't. So I was actually just talking to Agustina, and I don't even use ChatGPT right now, right? I am a mediocre computational designer compared to some of my colleagues in GPT, right? So my technical expertise is actually as a chemist and a biologist, right? So, so for me to present at the symposium, I think is very interesting because, these structures are they're bacterial, but they are very synthetic, right? They're very weird space age creatures, right? So and so I think there are parallels there, but whether we start to use some of the AI tools in this process, I don't know, because so much of the work is actually based on very practical, hand-done lab work, right? I'm pipetting things by hand, and this, you can automate it. Ginkgo Bioworks automates all of their biological processes. They automate a lot of there's a lot of people working in computational biology where they kind of have to design proteins and stuff like this. So this is where AI is used in synthetic biology. Whether I'm going to use it in any of these tools, like, I don't want to design an organism yet because you actually can't take them outside. Like, there's a lot of rules about organism design that I do not want to deal with, right? And I'd rather just use the wild type species and figure out, can you make an environment for them to be okay in? And they'll probably be more robust than any of the engineered species, to be honest. Like an engineered species, usually there's like a lot of metabolic load to it, and so like you're making, like say I want the cyanobacteria to like biocement way harder, so that it doesn't take 10 years for my structure to harden. There's a biological price to that, which we do not know of, and then, and that biological price will probably be something that you can't, that the wild type organism is better at beating, if that makes more sense. Like, they'll take over, right? Yeah, I don't know if that answered it. Sorry.
Oh,
Andrea, thank you for the presentation of the work. I have a kind of a future conceptual question, and then I have some technical questions. The future conceptual question is the sort of ability of survival. Right? These systems currently, they're developed in a lab, and in some way you have to build an environment for the architecture to exist, right?
Yes.
So it's a more of a kind of a different kind of biologically synthetic species, right? Which requires a whole nother level of production of architecture to even survive. This is very precious. And so how do you think of that in context of the kind of larger problems of ecology, biology? So that's sort of one question.
Yeah.
And the other question is more about the biosynthesization. I'm wondering, is this particular bacteria the only one you're using for MICP? And how do you deal with the smell? As you took it out, was the urea processes that are they, like, once you install it? Is there, like, what's the kind of a time frame between the cementization and then kind of curing? And I'm just curious about actually the relationship between the wet lab and then.
So yeah, there's several ways of getting MICP, right? So the PCC, like, it's a cyanobacteria, and it doesn't use the urolytic pathway, right? So S. pastori uses urolytic pathway, so that's the one that will make the ammonia smell, and that it's a very fast biocementer. PCC is like photosynthetic, right? So the biocementation comes as a byproduct. There's no urea based, and so there's no smell. But the
Do you mix it, or do you dip it? Is it a valve-based subdivision?
So it is. We put the bacteria in our binder, right, and then we feed it, I guess, with the media, and so, like, you can see they're very different, I guess, right? And so, yeah, I have used S. pastori, and like it makes me gag, but it's used in industry because it's quite effective, right? So my colleague, she has an exhibit at the Biennale as well, Karen, and she uses this bacteria, and it's much faster, and so there are a lot of advantages to it. This is the one that is used for soil consolidation because it naturally occurs in farms near where animals piss, right? And they use the urine to like consolidate the soil. So that's for the technical stuff. The question about, making this kind of synthetic environment, and like completely artificial in order to house this organism in a situation where it normally doesn't happen, is the more, not troubling, but this is the more challenging question where we did this show, and like I was just like, there's a lot of the pieces that don't work, right? They don't biocement because it's such a slow process. And so, yeah, but that's part of the test, right? Where we're trying to like take this outside for the first time, try to make an artificial structure, try to force ourselves to design our environment to accommodate something other than us, right? Which is quite challenging, I think. And then still respect the heritage structure that we're in, right? Yeah, and so what, like if I did not have the access to the resources I have at ETH, you know, this would be a very different project, right? So this, like the amount of money that ETH put into this was ridiculous. So if I had to do this in a university in Canada where there is no digital fabrication program, yeah, it would be based on, I think, much, like, I think it'd be closer to like an agriculture project, right? Where you're growing larger scale objects rather than kind of redefining an object from like the get-go, where I'm like, okay, I'm birthing an object from like these tiny bacterias or something, instead of trying to just grow plants into a shape or something like this, if that makes sense. Yeah.
Sorry. Yeah. Thanks a lot, Andrea. Hello. Hello. Hello.
Yeah.
Yeah. Thanks a lot, Andrea. I've become a huge fan. I saw the pavilion as well. It looks fantastic. Everything you do, it's just beautiful. I'm no judge of these kind of things, but it's clear that you know you can sell it well enough to make me think so, at least. And I also find it kind of beautiful, your candid take on, you don't even use ChatGPT, it's just good old lab work.
Yeah.
And then I'm really thinking about, just these kind of quite superficial differentiations or distinctions that we make between what is natural and what is artificial, right? So I think for us, it's kind of a flow in and out between things, and to even think of architecture itself as an artificial intelligence, because it's clearly synthetic, it is intelligent, and then you try to somewhat articulate things out of it. You have to externalize it, so it's no longer kind of human. It becomes artificial, and it acquires a new life on its own. Whether it remains artificial or natural, that's nobody knows. But then, so my question is more related to something that you didn't mention. I'm really curious about how you shape these things. The way things look, because there's a very particular aesthetics to it. And it's not just you, at your chair as well, but this kind of aesthetics is there, and somehow they kind of come together. You people have come together.
Well, there's an issue with us, right?
Yeah.
Like, so we
No, but I'm just curious about how do you go about designing this? Like, what are the kind of precedents in your mind? What are the discussions going on around these things? Like, what did you look at that you were like, okay, this should look like this?
Yeah.
Kind of pure intuition? Because
It's not pure intuition. I guess there's like a lot of reference. If you look at our Miro boards, there's like a lot of pictures of aliens or something, right? And so there was already a bias towards it. I could have made them as like very rational. They're all lattice structures, so I could have made them as like space trusses or something like this, right? And this was actually an option that we were discussing, but we took the forms of the two trees that were stuck in, that are kind of captured by the pavilion as reference points instead, and then tried to make these like trunks out of them, or trunks in reference to them. But yeah, it's something that I've been trying to reconcile in my own head, where it's like, why did it have to look like a bunch of space age, weird sea creatures or something like this, when it could have been much more simple? And part of it is I don't think in natural systems anything is like so rational like that, like as rational as we would design a bridge or something like this. Because I think there's an aesthetic to industrial efficiency, and then there's an aesthetic to biological resilience, and they are not the same, right? Like biological resilience is based on redundancy, like weak elements, things like, yeah, having multiple backups and stuff like this, and having flaws. Like there's a ton of flaws in these systems, right? Whereas industrial efficiency, these like very brittle systems that are so optimized, that comes with like a very particular aesthetic, I think, comes out of it. And so I was leaning towards the other side, right? As part of our design kind of impetus. Yeah.
So, hi Andrea. Again. So, I want to ask you something more architecturally because that's my biggest critique with ETH and DBT, especially. They do not never relate with the architectural spaces. And I saw your pavilion, and it looks like sculptures. Yeah. And this is a Biennale of Architecture. Do you want to habitate? It is possible to habitate these structures or not? Or do you think in the future maybe this is instead of being sculptures could be the columns of a big roof or something more habitated? Do you think it's possible? Or do you think
This material, no. Like this is not strong enough. Like unless you want to wait like 100 years for something to harden, right? This is not a structural material, and I'm not saying it's ever going to be a structural material. We chose not to make the architectural component because the research is not there yet. Like I can't integrate this into a building facade yet, right? The attachment part is quite challenging. And so like the material, if I was going to actually do like a building skin, I would probably use what Josh is made out of, right? But this is extremely expensive. Like some of these cross-linkable hydrogels that we use, it's like 500 francs for like a gram, right? So this is not feasible for an architectural biennale, right? So the materials that I'm using for these are much cheaper, and I can, like, this is part of the equation where, and I will say, doing a giant wall of this would have been insane, like in terms of the amount of labor and resources that it would have taken, right? So this is another part where architectural materials are cheap, right? So like living materials, because I'm doing it in a wet lab, this is like, I think people don't understand like running a biology lab versus running like an architecture group. The costs are not comparable, right? So like the amount of equipment that we need is very expensive, and because I'm doing much larger scale work than all of my colleagues in material science and stuff, my incubators cost like 6 times the price of theirs, right? So the things like this become an issue.
And do you think this kind of methodology, that's my second question, this kind of methodology, like right now you are showing how nature is so powerful, and we just ignored the nature in the last years. So we were just building with synthetic or artificial materials, and now you're showing this. But do you think there is a way to do this a little bit more affordable?
Eventually, yes. Like the reason this material is so expensive is not because there's anything particularly precious about that material. It's just it's not produced in the scale that we would expect as an architect, right? So like nothing of these materials is individually of the components is expensive. It's just like the synthesis is quite laborious until you optimize it, right? Like, and so I think most of the work is, looking at different processes to try to say we could probably do this if there was effort to make this at scale. Although, this is also like a big question for me, where the scale of production that we want to kind of reach as an architect sometimes is antithetical to having a healthy biological system, right? So, yeah, this is a whole other thing.
Hi, Andrea. Well, thank you for presenting. I was fascinated by the amount of working people that needed to be put into this project. And this is part of the comment and part of the question. And because we are in the playing models, and we had this dialogue of artificial intelligence and storytelling and architecture, when you were presenting, and with yesterday's presentation from my professor Alexandra, she had this spectrum of we can come from materials to, or we can go to geometry to robots. And then if we think of the same spectrum in geometry, you can have a specific intelligence. It can come either from the form itself or from the material, and then we can call it like low intensity. And then you go to the other spectrum where you have the robots, where you can say this is high intensity, and it's not about geometry nor material, but it's about circuits and your computation. So if we're in the same, what I want to say is that how I read through my lenses of computation and artificial intelligence is that the work you've presented is a collaboration between this low intensity, material driven intelligence with a high intensity, circuit driven intelligence, where you can see through the robotic procedure. So one of your answers were about I'm not computationally savvy. I don't think that, but I do believe that here in the whole process there is this interplay of intelligence, that being human intelligence, material intelligence, and then you have the hardware intelligence. So I really I think there is of course a lot of discourse of how much we can push further, and I saw on your slide as well that they say how much can we, there was one question there, how much can we go and for what and why? And I think you're also in the same state of mind that we put so much money into this, we had to create this synthetic environment, it needed to live there. That maybe the next step, and probably this could become a question, is what is next that you have engineered and you have solved all of these very technical problems that, and I was asking the bio cementing, the all the different chemical process and bacteria that you've already selected. How do you, what's next? Because probably geometry or columns or pavilions or stuff
what's next actually is more related to this, right? Where it's this is a very artificial system. We used a monoculture, a single bacterium, stuck it in a print and are trying to support it. And we do this because this is how science education is premised, right? In order to publish in Nature Communications, you have to have supporting documents that this is doing exactly this, and we proved this is how much CO2 it sequestered, and it's not for any other reason. Nothing like this will ever happen in nature, right? Nature is an ecosystem, and so there's millions of organisms working together. They're different species, and we do not know how they're cooperating together as a network, right? And so the biologists I work with, are quite interested in could you actually replicate, or could you house a community of organisms in the system, which I think I can. But what they're doing, we won't be able to parse out very easily, right? Because it might be one species that is maybe only 0.1% of the population in the entire communicate that is doing something that augments the activity of PCC, right? So things like that, which is, I think, much more interesting for us in terms of the technical work, right? But there isn't a lot of work in co-culture systems we're only starting to do that, and then you have to publish. You can have to have two species together, which also is like completely absurd in nature. Like you won't just have two species, but this is what we have to do step by step in order to publish places, and also for us to like, with our limited tools, to understand what's happening, right? Yeah, in terms of, yeah, so this is kind of the work where I want it to head, where just like, okay, can we start to like have a more holistic system that might not require as much artificial help, I guess, if that makes sense.
I'm just curious that this narrative of monoculture is actually really interesting, right? Because in some way you create a completely reductive ecological system that it's and you're sort of putting it in isolation, and then you try to kind of recreate, feed it when it starts strengthening when you feed it, right? So like how do you when you feed it to kind of activate the cementization process, how that's a biological problem in some way. So I'm curious, when you're and you're using the combination of the print with bacteria or the bath, right? Is that the moment of activation? Can you start seeing the kind of creation of the environment as an activation of that process, as almost like the fabrication is in designing of the room, right? Rather than sort of the creation of the kind of super hyper control of the monoculture.
Yeah, like part of it is like, yeah, using the room as a way of templating activity, right? Of like manipulating activity, which is not a nice word to use for like another life form, but this is part of it. It's like, why am I using heat mats and like the incubation chambers and stuff like this? It's so that we can encourage a certain process to happen, right? Yeah.
My question, thank you, Andrea, for this super, and I love that it's kind of, it feels like a kind of like a Donna Haraway SF kind of like got real in your work. So it's kind of like a science fiction, but it's not written, it is kind of made as a fabrication, it's like a science, yeah, that I find it super interesting and inspiring. I was wondering, like, do you think that this kind of work in a site like in the Venice Biennale, is that the kind of main site for this kind of work? Because there you have the kind of outreach and the kind of discussion almost as a kind of communication that you start to have with the visitors of the piece. Or could you imagine this also being in a context that is not in a, let's say, educational or showcase exhibition kind of setting, where it's more like a quiet bystander and it's just getting people out and they're like passing by.
Yeah, I mean, we wanted to just like put some in Zurich Lake, right, and see, because there's a lot of minerals in the lake that would help with the biocementation process, and for an audience of no one, right? Like the decay project I showed that goes back in the forest, we went to a lot of effort to set those things up, but it's for no one. Like it's on an isolated farm, and like we made it pretty just to like do that, and then and they're just kind of rotting there now. And so like a lot of, I think the effort is actually doing things not necessarily for the spectacle for people, but just because, I guess. I don't know if that's a good enough reason. But if we did do it in Zurich Lake, it's also because we want to see if it can survive, right? Yeah.
Any other questions? Comments?
short question. So, from my understanding, the intention behind using bacteria as a building block is the benefit of the sequestration of the carbon itself by the bacteria. Would you consider that in a hypothetical future where the material is sustainable without the constant need for attention, that the benefit of that carbon emission benefit, would it offset the ecological impact that the harvesting of the bacteria and the production has?
The harvest, I mean, the growing of the bacteria is not too energy intensive, right? It's just salt water and light. That's the thing. So I like working with the photosynthetic system because it's like a low intensity system versus the urolytic bacteria, which is, a powerful biocementer, but requires way more food, and has like way more byproducts, right? And so, even though the photosynthetic system is finicky in that it's slow, it's actually for me much lower maintenance than S. pastori. And whether it offsets like the amount of energy needed to kind of do all the fabrication, I guess, like the embodied carbon of the making process. This is something that we haven't researched yet, but there is evidence, like I showed the stromatolites, right? And these were the things that changed, like that geoengineered the planet, right? And they did it without a ton of, they did it with no human intervention, they just did it, right? So I think that's what I find interesting, that this family of bacteria is able to do that just on its own, right? So, yeah. Okay.
All right, so if there are no questions, I think we're done. So thanks, Andrea.